A simple question. I missed the distinction between the training set and the testing set. You have not given the specific numbers of patients.
So it was 100 in the test and 191 in the training. Thanks.
So did you, while we're waiting, see if anybody else has a question, did you consider the proteins in your survival outcome? Because we published, in the meantime, several manuscripts looking at individual proteins that are quite prognostic in this group for outcome, but neither of you guys used the proteins.
Yeah. We tried very hard, actually, to include the proteins. We used a number of methods. We used variable importance measures with random forest. We tried transforming it, principal components, and we didn't see any significant gains. It's very possible that there were gains, but we couldn't see anything beyond what potentially could have been random variation.
Same answer.
Actually, I would like to ask a question to Steve. Can I to understand the data? I was thinking, why is flu HDAC coming as a strong predictor? So, because I know a little bit about AML.
Piece of cake.
Okay, yes, exactly. I have some comments.
So the question was, why does fludarabine HiDAC, high-dose RSG, predict so well? So we started using fludarabine high-dose RSG in 1988 or 89, and it was initially a regimen that we used for unfavorable prognosis patients. But we then did a trial, a 4-arm trial, and we also used it in all patients. And the group that did the best with it was the favorable prognosis cytogenetic group. So for the time period where this data set comes from, the only people going on fludarabine HiDAC are favorable cytogenetic patients, with a couple rare exceptions. So it was a de facto marker for good prognosis cytogenetics. That's why most of the models don't have both 821 or inversion 16 and the fludarabine HiDAC, because they so tightly cross-correlate. That's also why for the subsequent challenge that's ongoing now, we've removed therapy. So you know, unfortunately, we didn't realize that before, and I think it drove the system too much so that people were looking at therapy. And I kind of wanted people to take advantage of the data that I have as a clinician on day 0 before I treat them, which would not include which therapy they got. But we didn't set it up that way initially.
Essentially, you don't think that should be the new standard therapy. It's just because the data were biased by this choice. But because I believe the standard therapy is adarubicin, cytarabine for AML.
Yeah, so the standard of therapy is anthracycline and RSG. And you can argue, and there's been great argument over 30 years of whether high-dose RSG is better or not. We'll leave that aside. And for the favorable prognosis patients, we use fludarabine high-dose RSG, and we get very good results with them. Other places will use anthracycline high-dose RSG. If I had done this study using samples from 1990 through 1998, as opposed to after that, you would have found the exact opposite, because we're using fludarabine high-dose RSG mostly for the poor prognosis patients, and you would have said it was a terrible regimen.
Thank you.
Witold Trudnicki, University of Warsaw. First, congratulations to my competitors. I fared quite poorly in this challenge, but I have a question or comment to the first presentation. As far as I understand, you actually didn't use this evolutionary information at all. Basically, this linear regression that we used with penalty didn't use this evolutionary information, because it was just the constant that you multiplied your linear, your variable by. So then this ridge regression simply, well, it didn't use this information from evolution. You have confirmed it, though, by because the variables that were selected had this high evolutionary conservation. But I think you didn't really use it in your actually in your methodology.
You may be right, because I really have no idea how that sparse logistic regression works.
What I did is I tried to multiply the z-score with the evolutionary weight. I thought after normalization with z-scores, everything was brought to a distribution centered at 0 with the standard deviation is 1. And once the evolutionary weight was applied, then they are no longer belongs to the original distribution. The distribution has been transformed in some way. And then this data set, when given to sparse logistic regression, must have some effect. But as to what specific effect it has, to be honest, I have no idea.
Maybe we can discuss it later.
Yes.
So I'd very much like to suggest that you do the subsequent competition, because it'd be very interesting to see the evolutionary conservation in the different subsets of patients. I think that might be more informative.
So I have one question for Stephen and one question or comment for David. David said that there was a batch effect. Can you recognize what was that batch effect in your?
Well, it was a single batch, so that's why I'm a little bit confused. I couldn't see what you were using to classify into what you said was red and blue, but kind of was washed out on the slide to me.
Yeah, so maybe, I will assert, if it didn't show in the figure, that there is a visible difference between 1 through 75 and through 76 through 199. I did things like go through individual genes and do t-tests to see if the means were significantly different. There were some genes, and I can't tell you which ones, I'm sorry, proteins, in that list that had really huge differences between the p-values. So I understand that visual inspection can be deceptive, so, but it would be interesting to take, the reason we mentioned it is it would be interesting to take a closer look.
One batch in RPPA, or because maybe the collection was different?
The collection was, but that's what I was going to ask is, Dave, I don't see you back. So can you tell me how you ordered them to get 1 to 75 and? Sorry?
It's the data as it happened. 1 to 75, the protein.
Yeah, so,
They were all normalized the same way using
yeah,
the current.
so I don't know,
Okay, we can
Yeah.
look at it. Too.
But they were also, they went through 4 different steps of normalization to get off the slide, so they were actually corrected in the same batch. So if there was anything like a spot on the slide, that was corrected for.
yeah.
I was saying there was a lot of rigor in actually getting the points, the data off of the slide into making sure that there wasn't a batch effect. So I would be surprised if there was, if you're not actually picking up real results from this protein array.
Yeah,
You know, maybe that cohort of patients was different or something too, but
I don't know. I don't know how Dave sorted, or, you know, I'd have to go back and see who those 75 are to see if there's.
so why don't we have a conversation on
Right.
offline? I mean,
Actually-
my main point is just that it would be interesting. In a real research situation, we would have had this conversation on day 2. Yeah.
But this is interesting that we will have at 6:30 a get together, and all the people who have worked on each of the challenges, you know, to discuss with each other in groups. So I hopefully that's one of the conversations that will be going on there. And the other question is for you. So did you guys finally? How did you use the RPPA data in the end?
In the end, we tried many different ways to use the RPPA data, but we ended up just excluding it all. We only used the 5 clinical covariates.
So I think that this brings the same problem that we saw in the RA challenge, that somehow the genetics or genomics is giving little information on top, if any, on top of the of the clinical data. And I think that's why we are trying to do the RPPA phase. But you know, Stephen, do you have any comment on that?
Definitely. So with the analysis that Wendy, wherever she is back there, raise your hand, there she is, hiding in the back, has done, we can find recognizable signatures. So if we do unbiased hierarchical clustering, we see somewhere between 7 and 9 signatures. And if we do a method that Wendy has a poster on, where we break things down into functionally related groups and then cluster based on which class you're in that, we see much more defined clustering within that. Now, to simplify it, if you have red leukemia and yellow leukemia and blue leukemia, and they have very different protein expression patterns, but you analyze them all together, go back to primary school where you got paint on your palette and you smeared it together and you always got brown, okay? And if you look for something in that brown, you can't see the red dots and the blue dots and the yellow dots. You've homogenized it by doing that. And so I think, I was hoping that the different teams would find better ways to classify the patients based on the protein, and that in combination with the clinical data would be more predictive. We have a lot of fairly striking things that we're seeing when we do it that way, big differences between them. And I think that the reason that it's lost in this subset, the reason perhaps that the RA subset or analysis didn't see benefit from the other things, is that there are different flavors, if you will, of the diseases, but you put them all together into one group for analysis, and you lose the ability to separate the different flavors or different colors.
So, you have another question?
Another rationale for the extended phase is because, again, a lot of the clinical attributes are not something that Steve or other clinicians can act on. So if you have a target or features which are predictive, which people can actually target, that's going to be much more clinically relevant. So that's one of the rationales for the extended phase.
Yeah.
Okay, so I think that if there are no more questions, then we finish the session. Thank you very much.